this on. Hello, everyone. My name is Joseph Boland from the National Cancer Institute of the United States. So today I really want to take you through the past probably year and a half since we've had the PGM and shortly the Proton. So let's get started. Okay, so as I said, I'm from the National Cancer Institute. Within the National Cancer Institute, obviously, there are several laboratories. My laboratory is the Cancer Genomic Research Laboratory. Currently, we're comprised of 40 scientists and bioinformaticists. 30 of those are on the production genotyping side. So my section of the laboratory deals primarily with sequencing. We're 10 people right now. And as you can see on the slide, we have one of every instrument in the market today, High-Seq, 454, and of course the PGMs. And that, oops, two things here. And this is actually one of the bays. And right now we have 6 PGMs, but we did start with 1. And so really I want to take you through what it was like when I had 1 and then the expansion into the next 6. So for today, as I mentioned, I just want to go through a historical review. So my section of the laboratory started 4 years ago. So at 4 years we had nothing. And we've kind of built it slowly. And as you know, this sequencing market, it's rapidly advancing. It seems month to month at this point. I want to take you through the first impressions of the PGM from the early days through some of the presentations we made at a couple of the conferences in the US. Go through what we have right now, which is the PGM is actually one of our main confirmatory pipeline instruments for our exome or familial exome pipeline, and I'll explain that further. Take you through the first transformation, so the first expansion, and that happened at Christmas of 11 when we decided to go from 1 to 6 PGMs. The second year with the PGM, we started to really push it. We knew we can amplicon sequence with no problem. Now we wanted to say, okay, let's start exome sequencing, and with the rapid development of their chips at Life, there really was no point to kind of lagging behind. We could actually push this thing further, and I have data to show you that the exome sequence we did on the PGM really foreshadows what's going to be done on the Proton. And then really want to talk about the future directions and then what we're calling the second expansion, and that's with the Protons that are arriving probably within the next month or two at our laboratory. So let's go all the way back to 2008. In 2008, I arrived at this laboratory. I had 1 tech and an empty room essentially, and tons of questions. Everybody wanted next-gen sequencing. No one knew how to do it at that point, at least in our laboratory, not in the industry. So we had tons of questions. We had investigators that wanted data the day before. We had several projects lined up. We had infrastructure items we had to deal with. And even today, when we bring in the PGM, it's not, it's almost plug and play, but there are some secondary items we need. We had to figure out what our staffing needs were. Was 1 person enough? The data storage, of course, over time that's now increased. And then, of course, the wet lab concerns with sample prep, sample quality. How do we quantitate the samples? How do you know you're putting 10 nanograms into an AmpliSeq study? And what confidence do you have? And then the bottom thing was to communicate between the bioinformaticists and the biologists. I'm a biologist, not a bioinformaticist, and that is a major problem in the US. I assume it's a major problem in Argentina. So that really needs to be explored. And you have to bring them in the laboratories, the computer people, as much as they hate it. You have to let them sit down next to you on the bench and really learn the technology of the science, and it really helps on the back end with the analysis. So in 2008, we had 1 sample flow. This is the good old days. We had 1 thing to do. Sample came in, sequenced it using the 454 process. And that was kind of nice, but we knew that wasn't enough. In terms of cost, the quality was very good, but cost per run was very high, and our throughput could be better at that point. So we had to keep looking for improvements to the process. In 2009, really what we focused on was the front end, was the sample prep end. So while we kept just 454 sequencing at that point, we really wanted to look at what was the way to go about sequencing, regional sequencing or amplicon sequencing. Was it through traditional PCR, or was it through the Nimble-Gin, CCAP, so basically the hybridization? And then we were looking at different ways to fragment our sample to get it into the process, whether it was the nebulization process or the sonication. And obviously the sonication won out due to its throughput and its ease of processing. But this was 2009, so for the most part, it was still a single sample flow. But with this industry and everything sort of exploding, this is what we looked like in 2010. So we go from one process to several. And so we have all these processes ranging from short read amplicons all the way to whole genome. Now, I should tell you that for the most part, we outsource our whole genome. We're not staffed for that as of today. And all these processes feed our three instruments, Ion Torrent and then the two competitors. But I think what we're going to see in 2013 or late 2012, what we really want to do is pare this back down, not lose any of the quality, not lose any of our ability or flexibility, but really pare down these processes and pare down the instrument that we feed. So we don't have to train so many people on so many different platforms. And I think what we're going to see is really we're going to start feeding just the Ion Torrent systems, with the Proton being able to handle your high-level exome, potentially whole genome, transcriptome. You can really start to see, or I can start to see, phasing out some of the other instruments that we have. So I've had the pleasure to give a variation of this talk a few times for Life, and it's been a wonderful collaboration dealing with the folks at Life. And really the first talk that I gave was a first impression talk about what it was like the first day we got the machine. So in November of 2010, we had our first conversations with Life about this instrument. And obviously we were interested from the start, but we did have a few things that we had to go through, a couple thinking points or talking points that we go through for all new technology in my laboratory. So we want to focus on 4 areas. Excuse me. We want to make sure that if we replace an instrument, we're not going to lose any quality. It's a very simple thing. These aren't major issues or major topics, but these are things that we hold true. So it has to maintain or exceed quality. We have to see a path to reduce costs. So we can take higher costs in the beginning as long as we see a path forward to lower costs. It has to be easy to use. With retraining, while we're doing projects at the same time, you don't have that kind of staffing level that I can take 2 people off a certain project and let them focus for 40 hours a week doing training. So this is really what, one of the key drivers is it easy to use. And of course, can we reduce project time and, by which we can do more projects with the given staffing level, or maybe a slight increase. And so, So just to get to the point, with the PGM, we've seen is all 4 of these have been easily met. Now, for ease of use, I focus on that. As a 454 user, it was a very easy transition to Ion. That's not to say that if you've never done anything, you can't do it. Actually, it's quite the opposite. It's very easy to adopt. Just for us, though, it was very easy to read those protocols and move forward. The quality issue was, in the beginning, as Andy said, it was about a 2% error rate over 100 base pair read. We are now at 150 to 200 base pair reads and a sub 1% error rate. So, we've maintained the quality of 454 and started to exceed it as well. The project costs have been significantly reduced using Ion. Just to give you somewhat of an idea, I was looking at a project last night that if we had run the same project we ran 2 years ago with Ion, we would have saved about 75% of our costs. And then, of course, project time. It used to be in the past, all projects would be months. You'd have to think in terms of months, 6 months. Now we're thinking about days and weeks. So really, it has been a transformative platform in our laboratory and the number of samples that we run through our lab. So the first month, so just to give you an idea, with the PGM, we purchased it in November of 2010. It arrived January 2nd, 2011. On January 4th, we ran our first samples. I've never had a technology that was up and running within 2 days. I'm not just saying that because I'm here representing Life, but it's actually the truth. So for the first month, really what we want to do, we had one goal in mind with the PGM. We knew that, at that time, it was going to be an amplicon sequencing platform. And we had several hundred samples that we can envision running through. So like everything we do in our laboratory has to be multiplexed. We have to have chips that can handle several samples at a time. We can't do one at a time. It's just too slow for us. So we took the barcode sets that we had at 454, and using a platform from Fluidigm, went through, and we wanted to challenge the system just to be able to differentiate the barcodes. So if we ran, as I did here in the first month, we ran a 1-plex. Could you tell me the barcode I ran? 8-plex, 24, and 48. And each time we would send the data to our bioinformatics folks, and then they would tell us which barcodes that they detected. And that's all we wanted to go after was the first month. Very simple, but very important at the same time. So this was what we wanted to do in the first month leading up to AGBT. Now, I'm going to reference AGBT a number of times. Some of you may or may not be aware. It's a conference in the US that for technology development folks, it's the Super Bowl of science. So every year we go, and it's always huge announcements. This year was Oxford Nanopore and the USB sequencer. I mean, it's always a big deal. So really what we wanted to do was be ready for that. So within the first month, we had 24 runs done.